Hello viewers, good evening and welcome to Ninja Peking. Do not forget to like and hit the subscribe button. On our last video, we talked about how the immune system helps in attacking or mounting its response against viral infection. And we talked about how the NK cells are being activated to release perforin and granzymes to do the killing in innate immunity. While in adaptive immunity, we talked about how the cytotoxic T cells were able to be activated and also do the killing by releasing perforins and granzymes. I also made mention of how we need memory for this reaction in case we are being attacked by the same virus. So today we're going to talk about how to create memory cells and also plasma cells that will be able to do the killing if eventually we do have this particular type of virus invading the host cell. And I had a viewer who sent a request to talk about antibody cellular dependent cytotoxicity. We'll go down there, but we have to know how everything goes about when cytotoxicity is all about. So from this video, he is going to understand what exactly antibody cellular cytotoxicity is, which is ADCC. Now, from our previous video, I told you a T helper cell is a form of CD4 cells, and they were able to differentiate by the activation of macrophages. They were able to differentiate to TH1, which activated the macrophage back to activate the pre-cytotoxic T cell that will now become a cytotoxic T cell to do killing. But in case of having a memory, it needs a memory cell, which is a B cell. A B cell is an example of a memory cell. It needs a B cell to be activated so that it can be able to save a memory for this type of attack against the same invading virus. So I am now going to give you a diagrammatic expression or explanation of how everything goes about. Okay, now this is our TH0 cell. It releases interleukin 19 and interleukin 23. It needs, before this occurs, before this occurs, it needs another T helper cell that will be able to activate the B cell to become memory cell. And the T helper cell that helps for memory is the TFH, which is the T follicular helper cell. T follicular helper cell. Please, on today's video, we're going to give key points. So the T cell, the T helper cell that is responsible for memory, for activating the B cell to have memory B cell, is the T follicular helper cell. I told you the T helper cell can be differentiated into TH0, TH1, TH1, TH2, T17, Treg, and TFH. So the one responsible for memory is the T follicular helper cell. So it needs a T follicular helper cell for the activation of a B cell to give us memory. So for these to occur, for these to be activated, the TH0 now differentiates to TFH by releasing IL-19 and IL-23 as the interleukin, the cytokine that it releases to differentiate, to make this activated so that it can activate the B cell. So when this happens, TFH also releases a self-stimulatory cytokine, the same cytokine, IL-12 and IL-23. On the body of the TFH, we have the CD40 ligand, CT40 ligand. So this is a self-stimulatory substance. So it means it undergoes clonal expansion. Go to our last video, I explained what clonal expansion is. So when this happens, it needs, I told you, it needs to activate a B cell. Do not forget, this B cell has also been infected by the same virus. So this B cell also is activated. Let me bring this here. CD40 ligand. So I'm just going to denote it with CD40L, CD40 ligand. And the body of the B cell, because it's an antigen presenting cell, and what do we say an antigen presenting cell is? An antigen presenting cell has both MHC1 and MHC2. MHC1 and MHC2. So we have the viral peptide here, and MHC1. Thank you. So now, the T follicular helper cell needs to bind to the B cell for it to be activated. The B cell, mind you, the B cell here is already activated because it's been infected by the viral peptide. But we need a memory cell. So how do we get the memory cell? This is what we're talking about here. Now, the T follicular helper cell now binds to the CD40 on the body of the B cell. On the body of the B cell, we have our T cell receptor mediated by the T cell receptor. Do not forget, this is also a type of a CD4 cell. So definitely this response is mediated by CD4. So when all these things are bound, the T follicular helper cell releases the same cytokine, IL-19 and IL-23. IL-19 and IL-23. So let's take a little recap. The TH0 helper cell needs a T follicular helper cell to be activated so that it can bind to the B cell, which has already been activated by the viral peptide, so that there's a bondage that allows the B cell to become a memory cell. So this is it. The T cell receptor binds to the MHC2 on the B cell, mediated by the CD4. The CD40 ligand binds to the CD40 on the B cell. And the T follicular helper cell releases IL-19 and IL-23 as the cytokine that activates the B cell to become memory cells. So beautiful. Memory cells. So mind you, it has MHC1 and MHC2. So now by so doing, the memory cell has both MHC1 and MHC2, showing the viral peptide, presenting the viral peptide to any humoral response, to any immune response, so that they can be able to code that this particular viral peptide has been fought. They know now the process of fighting this particular, so this particular memory B cell, it will go to your lymph, it will persist in your lymph for a long time, it will go into the bloodstream, persist in the bloodstream, so that whenever you have this invading virus, you have the immune mechanism of dealing with it, so it has a memory for it. So now, we want to explain antibody dependent cellular cytotoxicity. So when IL-19 and IL-23 is being released to the B cell goes into two parts. The first part is becoming a memory B cell. The second part, it becomes a plasma cell, which is what we all know as immunoglobulins, or antibodies. Beautiful. Immunoglobulins and antibody. Now let's take a little recap again. I need a T follicular helper cell to be activated so that it can bind to the B cell, so that the B cell can be activated, which is already activated by the viral peptide to become a memory B cell, and a plasma cell by releasing IL-19 and IL-23 from the TFH into the B cell so that it can become a memory cell and a plasma cell, which we all know is called immunoglobulins and antibodies. So what does this plasma cell do? And do not forget, immunoglobulins, we have 5 different types of immunoglobulins. We have IgA, we have IgG, we have IgM, we have IgD, and we have IgE. A stands for alpha, G stands for gamma, M stands for mu, D stands for delta, and E epsilon. Gamma and epsilon. These are the 5 different types of immunoglobulin we have. So what does this antibody do? What does it do? Now let me bring you back here. What does it do? What does the plasma cell do? What does it do? So I'm going to draw my antibodies this way. So these are plasma cells. Plasma cells. They go and find in the bloodstream, because it's in the bloodstream, they go and find the same viral peptide. They go and find the same viral peptide and code it, which is called neutralization. It goes into the bloodstream, find this viral peptide, and code it in a process called neutralization. Neutralization in immunology is defined as the process whereby neutralizing antibody are able to stop the infection by attacking the viral peptide, stopping it from binding to a particular cell to cause the infection. There are different ways it does this, by changing or stopping the conformational change of the viral peptide so that it cannot be able to bind itself to the cell it wants to affect. This is the process of neutralization. It neutralizes the viral peptides from binding to the cell, so it takes care of it. A question can equally come this way. Can neutralization be done for extracellular organisms? That means extracellular virus. A virus that is not found inside the cell? No. The immune system have a way of dealing with that type of particular problem when it comes to extracellular virus. E.g., it happens to use the process of opsonization and complement fixation to deal with that. But in this case, the virus is inside the cell, so it's called intracellular. So it uses neutralization reaction to take care of this virus. It coats the body, because it coats the body and neutralizes it so that it does not affect or infect a normal cell. 2. What does immunoglobulin do again? It's very beautiful. I want you to pay attention to what it does again. This is really cool. Really, really cool. This is the second thing it does. On the body of an infected cell, we have the viral peptide lining on the cell membrane of this infected cell. We have the viral peptide because it's infected by the virus, so we have the viral peptide on the cell membrane of the infected cell. So the immunoglobulin goes and attaches itself to the viral peptide found on the body of the cell membrane. So do not forget, on the body of the antibody, we have the Fab, which is the binding site of the antigen. So on the body of the immunoglobulin, we have the Fab region, which is the binding side of the antigen. So this is the particular position on the antibody's body, on the antibody that binds to the antigen, the Fab region. Then on the immunoglobulin or antibody, we have also the FC region. So when the immunoglobulin binds to this, to the infected cell, there is a particular T cell that recognizes this bond between the immunoglobulin and the infected cell, which is our NK cell. Again, do not forget NK cell. It recognizes this. It recognizes this bond, the binding of the infected cell and the immunoglobulin. Now look at this. Look at how it will do the killing. Look at how it will do the killing. Okay? The NK cell, on the body of the NK cell, there is a portion on the body of the NK cell that attaches itself to the FC region of the immunoglobulin. A receptor. A receptor. This receptor is called FC gamma 3. Gamma for immunoglobulin G. Another name for FC gamma 3 is also called CD16. And do not forget, on the body of the infected cell, on the NK cell, we have the CD40 ligand. On the body of the infected cell, we have the CD40. So when all these things are bound, the NK cell now releases, the NK cell now releases the same protein, perforin, protease, yes, and granzymes to do the killing. And what do we call the killing? Cell apoptosis or cell death. This process in which the NK cell recognizes the bond between the immunoglobulin and the infected cells and binds its region, the FC gamma 3 or CD16, so that it can bind and release perforins and granzymes, is called antibody-dependent cellular cytotoxicity, because it's dependent on the antibody to do the cellular killing. Antibody-dependent cellular cytotoxicity. So it is dependent on the antibody to do the killing. The NK cell is dependent on the antibody to do the killing. And how is it dependent? Let's take a recap. The H0 differentiates into different types of T helper cell, but it needs a T follicular cell to have a memory where it can actually activate the activated B cell to become a memory B cell and a plasma cell. The plasma cell goes on and undergo a process called neutralization reaction by neutralizing, by coding the viral peptide found in the bloodstream and also in the cell, and also allows it to bind to an infected cell through the FC region by triggering the NK cell, recognizing this bond between the infected cell and the immunoglobulin, releasing perforin and granzymes. This process is called antibody dependent cellular cytotoxicity, which is ADCC. I hope I tried as much as possible to answer your question. If you have more questions on topics like this, please you can always drop them on the comments forum. Thank you very much and have a wonderful day.